For Psychopharm Archive, I am a consultation liaison psychiatrist and academic physician. If you value this high-level analysis of medical history and neurochemistry, subscribe for our next deep dive into the molecules that shaped the modern world. In January 2009, the United States Department of Justice finalized a resolution that served as a defining moment in the history of pharmaceutical oversight. Eli Lilly and Company agreed to a $1.415 billion settlement to resolve criminal and civil allegations regarding the off-label promotion of Olanzapine, a thienobenzodiazepine derivative commercially known as Zyprexa. While this figure was eventually surpassed by the $2.2 billion Johnson & Johnson settlement for Risperdal in 2013, the Zyprexa case remains scientifically and ethically significant because of the nature of the data involved. At the time of the announcement, the $515 million criminal fine was the largest ever imposed on a single corporation in United States history. The litigation stemmed from a strategic marketing initiative known internally as the Zyprexa Limitless campaign. As documented in a 2006 New York Times investigation based on leaked internal documents, commonly referred to as the Lilly Papers, the company engaged in a systematic effort to promote the drug to primary care physicians for conditions far beyond its FDA-approved indications. This included targeting geriatric populations for dementia-related psychosis and pediatric patients for various behavioral dysregulations. The controversy was not merely a matter of regulatory overreach, but a fundamental conflict regarding the disclosure of metabolic attrition. Internal communications, such as those highlighted in a 2009 analysis by Greg Spielmans, revealed that by 1999, internal researchers had identified a substantial risk for weight gain and hyperglycemia. Despite these internal findings, external marketing continued to emphasize a superior tolerability profile, particularly regarding the absence of extrapyramidal symptoms common in first-generation agents. This institutional history creates a striking paradox. Despite the multi-billion-dollar legal fallout and the well-documented suppression of metabolic risk data, Zyprexa has maintained its status as a cornerstone of clinical psychiatry. It is frequently the agent of choice for treatment-resistant cases and acute stabilization. In this archival review, we will move beyond the headlines to examine the molecular architecture that grants this molecule its clinical superiority. We will analyze the binding affinities and dissociation kinetics that differentiate it from other atypical antipsychotics, reevaluate the outcomes of the clinical antipsychotic trials of intervention effectiveness, or the CATIE trial, and integrate emerging data from a late 2025 meta-analysis regarding long-term oncogenic potential and prolactin-mediated risks. The objective is to reconcile how a molecule with such a fraught corporate history remains an indispensable tool for the management of severe psychopathology. As of early 2026, the clinical utility of olanzapine is codified by a broad range of FDA-approved indications. It remains a primary treatment for schizophrenia in both adults and adolescents aged 13 and older, and for the acute and maintenance treatment of manic or mixed episodes associated with bipolar I disorder. In combination with fluoxetine, the formulation known as Symbyax, it is indicated for treatment-resistant depression in adults and for depressive episodes associated with bipolar I in patients as young as 10 years old. Most notably, the recent introduction of the olanzapine and samidorphan combination, marketed as Lybalvi, represents the latest regulatory effort to maintain the molecule's efficacy while mitigating the metabolic attrition that has historically limited its long-term use. The clinical profile of olanzapine is defined by its complex polypharmacy at the receptor level. Its pharmacological architecture is characterized by a high affinity for multiple neurotransmitter systems, which accounts for both its robust efficacy and its significant metabolic liability. When examining the binding constants, or Ki values, we see a distinct hierarchy of receptor occupancy. Olanzapine demonstrates a potent antagonism at the serotonin 5-HT2A receptor with a Ki of approximately 4 nanomolar. This is contrasted with its affinity for the dopamine D2 receptor, where it exhibits a Ki of roughly 11 nanomolar. This favorable 5-HT2A to D2 ratio is a hallmark of atypicality, facilitating the disinhibition of dopamine in the prefrontal cortex while maintaining sufficient blockade in the mesolimbic pathway to address positive symptoms. Furthermore, the molecule's interaction with the D2 receptor is characterized by loose binding kinetics. As discussed in the foundational 2001 research by Kapur and Seaman, olanzapine dissociates from the D2 receptor more rapidly than first-generation antipsychotics like haloperidol. This transient occupancy allows for sufficient endogenous dopamine signaling to minimize the risk of extrapyramidal side effects and tardive dyskinesia, even at doses required for acute stabilization. However, the molecule's efficacy is inseparable from its high affinity for other sites. It possesses an extremely high affinity for the histamine H1 and muscarinic M1 receptors, with Ki values near 1.9 nanomolar. The potent H1 antagonism is the primary driver of the rapid onset sedation and hyperphagia observed in clinical practice. Simultaneously, the M1 antagonism provides an intrinsic anticholinergic effect that further reduces the risk of motor side effects, but contributes to the cognitive blurring and autonomic changes frequently reported by patients. This shotgun approach to receptor binding creates a unique clinical footprint, a highly effective antipsychotic that necessitates a rigorous trade-off with metabolic and cognitive health. To understand why clinicians continue to accept this trade-off, we must examine the landmark clinical trials that established olanzapine as a superior agent in terms of treatment retention. The most significant of these is the Clinical Antipsychotic Trials of Intervention Effectiveness, or the CATIE trial, published in the New England Journal of Medicine in 2005. In this massive Phase 1 double-blind study, olanzapine was compared against other second-generation antipsychotics including Risperdal, Seroquel, and Geodon, as well as the first-generation agent perfenazine. The primary outcome was time to treatment discontinuation for any cause, a metric designed to reflect both efficacy and tolerability. The results were definitive. Olanzapine demonstrated a significantly longer median time to discontinuation at 9.2 months, compared to a range of 3.5 to 5.6 months for the other agents. Patients remained on olanzapine longer because it was more effective at preventing relapse and psychiatric hospitalization. However, this success was paradoxical. While it outperformed its peers in efficacy, olanzapine also had the highest rate of discontinuation, specifically due to weight gain and metabolic effects. This creates the central tension in the use of this molecule. It is the agent most likely to keep a patient stable, yet it is also the agent most likely to induce significant physiological morbidity over the long term. This finding from the CATIE trial mirrors earlier data from the original 1996 Beasley papers, which established olanzapine's broad-spectrum efficacy across positive, negative, and depressive symptoms, but warned of the metabolic costs that would eventually lead to the massive litigation of the following decade. The litigation of the 2000s did not diminish the molecule's standing because its clinical superiority is repeatedly validated by the highest hierarchy of evidence. In a landmark network meta-analysis published in The Lancet by Leucht and colleagues, which analyzed over 400 randomized controlled trials of 32 antipsychotics, olanzapine consistently ranked in the highest tier for overall symptom reduction. The data positioned the molecule as second only to Clozaril and amisulpride in terms of its standardized mean difference in efficacy. This statistical dominance is further supported by a 2019 analysis in JAMA Psychiatry, which confirmed that olanzapine's impact extends beyond positive symptoms to a statistically significant effect on negative and depressive symptoms, a multidimensional profile that few other atypical agents can empirically sustain. This efficacy is reinforced by real-world effectiveness data from massive national registries in Finland and Sweden. Longitudinal studies led by the Jari Tihonen Research Group, with data updated through early 2026, have followed tens of thousands of patients for over a decade. These registers consistently demonstrate that olanzapine is associated with one of the lowest risks of psychiatric rehospitalization and all-cause mortality among all oral antipsychotics. In these naturalistic settings, effectiveness is measured not just by a point reduction on a scale like the PANSS or BPRS, but by the prevention of relapse and the preservation of life. This suggests that the stabilization provided by the molecule's complex receptor architecture translates into a measurable survival benefit that, in the most severe cases, outweighs the documented metabolic liabilities. Furthermore, high-impact research published in early 2026 has reevaluated the drug's role in treatment-resistant cases. A systematic review of 12 studies comparing high-dose olanzapine, defined as 20 milligrams per day or greater, directly against Clozaril found no statistically significant difference between the two molecules in terms of overall psychopathology or negative symptom reduction. While Clozaril maintained a narrow margin for positive symptoms, high-dose olanzapine emerged as a statistically comparable and viable alternative for clinicians managing refractory cases where Clozaril is contraindicated. This robust evidence base from reputable, high-impact journals explains why, despite the history of the Lilly papers and the multi-billion-dollar settlements, Olanzapine remains a first-line necessity in the management of complex psychopathology. The objective of this archival review is to reconcile the drug's clinical indispensability with its profound biological cost. To do so, we must move from the statistical findings of the CATIE trial to the precise molecular mechanisms of metabolic attrition. The hyperphagic response, the rapid and often uncontrollable increase in caloric intake, is not merely a behavioral side effect, but a direct disruption of the hypothalamic energy balance circuitry. Research published in Psychoneuroendocrinology has identified that olanzapine acts as a potent antagonist at the histamine H1 receptor, which in turn inhibits the interaction between the H1 receptor and the ghrelin receptor, or GHSR1A, within hypothalamic neurons. This molecular disruption triggers the activation of AMP-activated protein kinase, or AMPK, and subsequently increases the expression of neuropeptide Y. By activating the PFOXO1 and PCREB transcription factors, olanzapine effectively forces the hypothalamus into a state of perceived energy deficit. The resulting orexigenic signaling bypasses standard satiety cues, explaining the significant and rapid weight gain that led to the early litigation. However, the metabolic liability of olanzapine extends beyond weight gain to a direct impairment of glucose homeostasis. High-impact studies have demonstrated that the molecule induces apoptosis in insulin-secreting pancreatic beta cells through a unique disruption of the endoplasmic reticulum stress response. Unlike other second-generation antipsychotics, olanzapine blocks the P-E-R-K-mediated translational attenuation pathway. While it activates the P-E-R-K sensor, it paradoxically prevents the phosphorylation of the alpha subunit of eukaryotic initiation factor 2, or EIF2-alpha. As a result, protein synthesis continues unabated despite high levels of cellular stress. This leads to an intracellular accumulation of misfolded proinsulin within the endoplasmic reticulum, triggering unresolved stress and, ultimately, programmed cell death. This explains the clinical phenomenon of rapid-onset hyperglycemia and diabetic ketoacidosis in patients who have not yet developed significant adiposity. It confirms that the metabolic attrition identified in the Lilly papers is a multi-organ failure of cellular homeostasis, providing the biological substrate for the discontinuation rates observed in the KAYT study. The long-term safety profile has been further refined by a landmark meta-analysis published in early 2026. The Karimi et al. Study, which pooled data from 13 observational cohorts, confirmed a 23% increased risk of breast cancer in females treated with antipsychotics. For olanzapine, a mid to high prolactin-inducing agent, the risk is particularly pronounced in long-term treatment. Epidemiological data indicates that cumulative doses exceeding 10,000 milligrams of olanzapine equivalent are associated with a hazard ratio of 1.29. This oncogenic risk is mediated by the chronic elevation of serum prolactin, a direct consequence of dopamine D2 blockade in the tuberoinfundibular pathway. Prolactin acts as a mitogen in mammary tissue, promoting the proliferation of epithelial cells and potentially accelerating the growth of subclinical malignant clones. As of March 2026, this data has fueled new large-scale litigations, alleging that the manufacturers failed to adequately warn clinicians of the tumorigenic signals identified in early rodent models, signals that were reportedly known as far back as 1996. While the 2026 litigations focus on the suppression of tumorigenic signals, they represent a recurring institutional pattern that defined the molecule's first decade of commercialization. The discrepancy between internal data and external promotion is a historical precedent that was first codified during the strategic marketing initiatives of the early 2000s. This era was defined by the Viva Zyprexa and Zyprexa Limitless campaigns, which focused on expanding the drug's use beyond its FDA-approved indications for schizophrenia and bipolar disorder. The objective was to penetrate the primary care market by encouraging 59,000 general practitioners to prescribe the molecule for mood, sleep, and anxiety, or MSA. These pervasive marketing tactics, which systematically prioritized market expansion over pharmacological transparency, eventually triggered a massive multi-state and federal investigation into the company's promotional practices. The culmination of that specific legal inquiry was the January 2009 settlement with the United States Department of Justice. Eli Lilly pleaded guilty to a criminal misdemeanor for misbranding the drug specifically for dementia-related psychosis in the elderly between 1999 and 2001. This resulted in a $515 million criminal fine, the largest corporate criminal penalty in United States history at that time, and an $800 million civil settlement. The DOJ investigation confirmed that the company had utilized a specialized long-term care sales force to promote the drug for mood stabilization in nursing homes, despite internal awareness of an increased risk of mortality and stroke in geriatric populations. Furthermore, the litigation exposed the strategic manipulation of medical literature. Documents revealed that the company's weight change narrative was a calculated effort to use neutral terminology in place of weight gain, despite clinical trial data showing that 30% of patients gained more than 22 pounds within 12 months of treatment. This systematic suppression of data regarding metabolic attrition was the central catalyst for over 18,000 individual product liability lawsuits, which the company eventually settled for over $700 million. For the archival record, the Olanzapine litigation stands as a definitive case study in how commercial limitless objectives can fundamentally conflict with the clinical requirement for informed consent and data transparency. In the final clinical calculus, olanzapine remains a drug of profound contradiction. Its superior receptor kinetics and loose binding at the dopamine D2 receptor provide a level of stabilization for treatment-resistant schizophrenia and bipolar disorder that few other ligands can approximate. Yet, as demonstrated throughout this archival review, this efficacy is inseparable from a biological cost that encompasses hypothalamic dysregulation, direct pancreatic beta cell toxicity, and a statistically significant long-term oncogenic risk identified in late 2025 data. For the modern practitioner, the utilization of this thienobenzodiazepine requires a rigorous, evidence-based trade-off, leveraging its unparalleled psychiatric effectiveness while maintaining a constant data-driven vigilance over the systemic physiological architecture it inevitably alters. For Psychopharm Archive, I am a consultation liaison psychiatrist and academic physician. If you value this high-level analysis of medical history and neurochemistry, subscribe for our next deep dive into the molecules that shaped the modern world.